So fine. If you remember, we were discussing something related to how the impulse which is generated by the action potential is carried throughout the length of the neuron to the axon terminus. And that happens by the way of certain messengers which are synthesized by the nerve cells, which we commonly refer as the neurotransmitters. And apart from these compounds, there is another way in which the neuronal impulse can be transmitted in a much faster way. And that happens in case of the neurons which are connected together by means of the electrical synapses. So, electrical synapses are basically very similar to what are referred to as tight junctions. And at the synapse, which is, say, this area, there are channels which are connecting the two membranes. That means the membrane of the axon terminals, which is a presynaptic neuronal membrane, to the membrane of the dendrites, which are going to form the synapse with the axon terminal, and that membrane is referred to as the postsynaptic neuronal membrane. So they are virtually very closely connected with each other in the electrical synapses. So what happens is that whenever the action potential, which is the depolarization of the presynaptic membrane, whenever the action potential reaches to the axon terminus, that action potential is directly relayed to change in the action potential of the postsynaptic neuronal membrane as well, and then it propagates all throughout the next neuron. So essentially, this kind of neuronal impulse transmission is referred, or it happens in electrical synapses, which are very, very fast transmitting neurons. However, many of the neurons, major of the neurons rather, connect with each other by means of what are called as chemical synapses. And chemical synapses are having a gap between the pre and the postsynaptic membrane, and they are called as chemical synapse because they actually uh, communicate with each other. That means the presynaptic neuronal membrane and the postsynaptic neuronal membrane communicate with each other by means of certain chemical signals, which we commonly refer as the neurotransmitters. So when we are talking about the chemical synapses, then the type of the neurotransmitter decides, as well as what kind of a receptor is harbored in the postsynaptic membrane, that decides that in what manner it is going to react to the neurotransmitter which is released by the presynaptic membrane. Is this much fine? Should we go ahead from this point?
Yes, sir.
Okay, so how this entire stuff happens, we took a glimpse of it in the last class, uh, where we discussed that how acetylcholine is actually responsible for bringing about a change in the membrane potential at the neuromuscular junction, and how that changed action potential changed sorry membrane potential is responsible for release of calcium ions from the sarcoplasmic reticulum. Calcium ions in turn are going to be responsible for triggering the action of myosin motor proteins, which are going to gain the activity of ATP hydrolysis in response to their binding with actin. Now, this binding of myosin with actin happens or is driven through calcium ions, because calcium ions bind to these regulatory proteins, especially troponin, and that leads to changes in the conformation, and thereby unmask the sites of myosin binding on the actin filament. And now myosin has an access to actin, it can bind to actin, and that activates the ATP hydrolyzing capacity of the myosin heads, which brings about the power stroke, and that leads to contraction of the muscle. That was one aspect. However, the other aspect associated with myosin protein is essentially related with a kind of myosin which is referred to as myosin V or myosin 5. Now, what this myosin does is quite a unique activity, and the activity which is associated with this myosin is responsible for transport of the synaptic vesicles, which are filled with neurotransmitters that are synthesized at the cell body of the neuronal cell. Once they are filled and they are packaged with the neurotransmitter, this synaptic vesicle needs to move all throughout the length of the axon and get concentrated at the axon terminus. So this is the area where it needs to get concentrated. Now, if it wants to move from this point to the end of the axon, it needs to be transported somehow, and that happens by means of the action of myosin V. This is an interesting protein as it also has the head groups, which are going to be bringing about what you can say the actin guided ATP hydrolysis in order to bring about the power stroke. Now, the arrangement is quite unique. Arrangement is on the synaptic vesicle. Now, if this is your synaptic vesicle, if you want to look at it like this, if this is the synaptic vesicle, then on the membrane of the synaptic vesicle, there are the myosin heads protruding out, just like some kind of legs are coming out from the synaptic vesicle. And now, when they are brought in contact with the actin filaments, which are lining the cytoskeletal arrangement of the axon, then the myosins can bind here and bring about a power stroke that actually makes this synaptic vesicle to roll over and get transported to the end of the axon at the axon terminus. Now, what happens at the axon terminus? That's our focus of the study for today's class. So I hope that till this point you are with me. And if there are any questions regarding uh, uh, this aspect, then we can discuss it. Or rather, in case you are good with the concept, then we can move ahead and understand that what is happening at the synapse. So what should
Sir,
we do?
can you please repeat this last topic?
What regarding myosin guided transport of
Yes, yes, sir.
synaptic vesicles? Okay, so this is an interesting protein which what you can say an alternative form of the myosin which is present in your muscles. This myosin 5 has this unique character that it gets embedded in the membrane of the synaptic vesicles. Now, synaptic vesicles are synthesized at the cell body. They are getting filled up with the neurotransmitter at the cell body. So they need to get transported to the axon terminus where their action is needed. That means they are going to release the neurotransmitter in the synapse. So how come these synaptic vesicles that are synthesized at the cell body get transported all throughout the length of the axon to the axon terminus? That is done by myosin 5. Now, there are other motor proteins as well which take part in this kind of movement. They are dynein-like proteins and they are associated with microtubules. That's a different aspect. But because you have already studied something about myosin, telling you something the aspect of myosin is easy for me. So I'm not focusing on other motor proteins. They are also involved, however, in the transport. Now, what happens is the myosin, which is coming out, or its heads are protruding out from the synaptic vesicle, these heads come in contact with the cytoskeletal network of actin, which is actually maintaining the shape of the axon. So, what happens is that this synaptic vesicle, whose myosin proteins are protruding out, will get docked here somewhere. And ultimately, what happens is that as soon as myosin makes contact with actin, it starts hydrolyzing ATP that generates a power stroke. So it actually causes a movement which is somehow oriented in this direction. So what will happen is that ultimately this synaptic vesicle will get transported here with the two head groups directed, while these two head groups are going to make the contact now with actin. and it will look somewhat like this. And this will continue to roll. So if you can imagine, it rolls down all throughout the length of the axon and gets to the axon terminals and get concentrated here at the axon terminus where it needs to show its action. This kind of transport of the synaptic vesicles from the cell body to the axon terminus is referred to as anterograde transport. Okay, there is another transport mechanism where the empty synaptic vesicles are transported back from the axon terminus to the cell body so that they can get refilled and they are transported back. So from axon terminus, if the empty synaptic vesicles are moving towards the cell body, this kind of transport is referred to as retrograde transport. So I hope that this much point is clear. Should we go ahead from this point now?
Yes, sir.
Okay, so the next step is where by hook or crook, say suppose you have got the synaptic vesicles here at the axon terminus. Now they need to execute their function. And how that happens, let's look at this point now. Okay, this is okay. I will come back to this slide just a bit later. We will talk about first what is happening at the synaptic cleft, and then perhaps you will understand that how the vesicles actually get empty at the synaptic terminal. So this is the synapse. This is the actual structure of the synapse. When the action potential arrives here, then by some or other way, it makes these vesicles to come in contact with the presynaptic membrane. Now, I hope that the nomenclature is clear in your mind. This is the presynaptic membrane. This area, and this is the postsynaptic membrane. Now, presynaptic membrane is the recipient of the action potential. That means it is coming from this end. Now, if you look at the neuron, it looks like that it is coming, the neuronal impulse is coming in this direction. That means the action potential arrives here. So it comes here at the axon terminus. This is the area, and this is the presynaptic membrane, which is enlarged over here for everyone's understanding. So as soon as the action potential arrives at the presynaptic membrane, by some or the other way, the synaptic vesicles are brought in contact, close contact with the presynaptic membrane. And now, as we know that all the vesicles are actually going to be having a phospholipid bilayer, and even the membrane has a phospholipid bilayer, and lipids tend to coalesce, that means tend to get mixed up together quite easily. Now, this is something like you have very fine oil drops, and now when you keep them quite close enough, then they merge together in order to form a big oil droplet. Uh, is this analogy good enough for you to understand that what is actually happening?
Yes, sir.
Yes, sir.
Okay. So now when the synaptic vesicle merges with the membrane, synaptical vesicular membrane merges with the presynaptic membrane, then entire neurotransmitter load is actually going to be released in the synaptic cleft. Now, as soon as you release the neurotransmitter in the synaptic cleft, then what is going to happen is the interesting part. Now, once it is released, the neurotransmitter searches for a receptor in the postsynaptic membrane. So now, if you have the receptor for this neurotransmitter in the postsynaptic membrane, what may happen? Now, the receptors can be of different types. We will talk about that later on. But when the neurotransmitter binds to some of the receptors, which are receptor coupled channels, you can consider like that. Okay, or you can consider them as the very commonly used term, ligand gated channels. Now we have seen voltage gated channels, right? Which in response to voltage change open. Ligand gated channel, ligand here is nothing but the neurotransmitter. Ligand gated channels are the ones which open in response to the neurotransmitter. Okay. So now just imagine that if it is a sodium ion channel, and now listen to this carefully, okay? And you need to answer this question, what I'm trying to ask. If the receptor in the postsynaptic membrane is a sodium ion ligand gated channel or a neurotransmitter dependent channel, which is going to open in response to binding of the neurotransmitter, then what's the fate of the next neuron? What will happen at the next neuron? Is the question clear? Yes?
So can you please repeat?
What? The question?
Yes, sir.
Okay. If the postsynaptic membrane has a sodium ion channel, which is ligand gated, and the ligand here is neurotransmitter, then in response to the neurotransmitter, what will happen? What will be the fate of the postsynaptic membrane? What will happen at the postsynaptic membrane? As you release the neurotransmitter will bind to a sodium ion ligand gated channel, which will open in response to the neurotransmitter. So what do you expect to happen in the postsynaptic neuronal at the postsynaptic neuronal membrane?
So action potential will generate.
Yes, obviously. So what do you think? Will the next neuron be firing in the same manner like the first neuron does? Yes. The action potential of the first neuron has arrived at the axonal terminus, or at the synapse, you can say, and that is actually opening ligand gated sodium ion channels in the postsynaptic membrane. So that will generate action potential in the postsynaptic membrane as well. So will this neuron fire, or will it not fire, according to you? Will there firing, meaning will there be another action potential that will be generated and will migrate towards the end of this neuron?
Yes, sir.
Yes, it will, right? So in the same manner, what is happening, the same kind of an impulse will get transported. So now, is this question becoming clear? Now, if you remember, I didn't tell you anything about how the action potential gets generated, right? Do you remember that? That means
Yes,
it
sir.
begins from the axon hillock. That's what I told you. But we didn't discuss that how it gets initiated there. It gets initiated in this manner. That means if the previous neuron fires and is in contact with the with the receiving neuron, then the receiving neuron, if it has these ligand gated sodium ion channels, then in response to the neurotransmitter, they are going to get open, and that will generate a second action potential in the second receiving neuron. Is this much fine?
Yes, sir.
Right. Now the question arises, how long this will continue? Is this a valid question to ask?
Yes, sir.
Okay, so what's your answer in this? How long will this process continue? Meaning, because this has released neurotransmitters here in the synaptic cleft, neurotransmitters are going to remain there for some period of time. So for that entire period, all the sodium ion channels will open here, and that will continue to generate action potential. So this neuron will continuously fire, or how long will it continue to fire? Yes. Don't you think that this is a valid question to ask? Because as long as the neurotransmitter stays there, it will continuously remain bound to the ligand gated channel, and it will keep that channel open, right? So more and more sodium ion will rush in, and more depolarization will take place. So this action potential will be a continuous process, right?
Yes, sir.
So do you want this process to be a continuous process, or do you want it to cease somewhere, or do you want that it should stop somewhere?
So it should stop.
Right. So how will you control that?
So there must be something which is controlling
Obviously.
or which is
So
closing the ion channel.
which is closing the ion channel. Now this doesn't happen, okay, because that's a slightly a different job to do. Because here it's not like the voltage gated channel. This is in response to the neurotransmitter. So it cannot afford to get into what is called as an inactive state, like the voltage gated channel, because more and more presence of neurotransmitter, when the guidance is given by the neurotransmitter here, it's not what is given by the voltage. So it's not like voltage gated channel that you can inactivate it. You rather cannot afford to inactivate it, because you want that till the neurotransmitter is there, there should be a continuous generation of action potential, right? So how will you control? There should be some ways that you can also think about. There is no nothing difficult in this to imagine. Yes.
Sir, if we stop the release of neurotransmitter by something.
uh, there is no such control available, rather, that you can stop the release of neurotransmitter. And then there needs some degree of intelligence there, right? That how long you should continue to release neurotransmitter, and how after what time you should stop releasing neurotransmitter.
Yes, sir.
So you cannot actually stop releasing neurotransmitter. So any other way of controlling it? It's actually in front of you. If you look at the screen carefully, then you will understand. Yes. Any idea?
No, sir.
Okay. No one has any idea about it? It's actually in front of you. You can see my cursor hovering over some area, right?
Yes, sir.
Okay. Does that give you some direction to think?
Sir, if the sodium ion channel closes,
Yes, but how will it close? Can anybody tell me what this compound is? This is nothing but this acetyl group. This is the choline unit. This is actually acetylcholine, the neurotransmitter, right? And How does it control? That means how is it its action is controlled? The receptor, either the receptor itself or there are certain enzymes, like for example, for acetylcholine, there is an enzyme which is called as acetylcholine esterase. Now, this is present in the synaptic cleft in this area. Okay. So, what happens is that it is always here. Now, acetylcholine esterase, I will write it like ACE. Okay. Now, what happens is that it is here at a particular concentration. And the neurotransmitters, as soon as we consider the neurotransmitter is acetylcholine now, as soon as they are released, there is always a competition between acetylcholine esterase and the receptor for binding of the neurotransmitter. Okay. So if it binds to acetylcholine, acetylcholine esterase breaks it down into two components. One is the acetate group, or you can consider acetic acid, and the other is choline. Okay. So it gets broken down into acetate and choline. So what happens is that if it is getting broken down into acetate and choline, it can no longer act as a neurotransmitter. However, if it binds, if it somehow escapes from the action of acetylcholinesterase and binds to the receptor protein, which is a ligand gated ion channel here in this case, then it opens the ion channel and creates the action. Now, what is the controlling factor here according to you?
Acetylcholine esterase.
How come? It's
So
not
because
only acetylcholinesterase. It is the proportion of acetylcholinesterase to the receptors. So if this
yes, sir.
balance gets disturbed, now just imagine if it is 1:1, then there is 50% chance that 50% of the neurotransmitter will bind to acetylcholinesterase and will get destroyed. And there is 50% chance that it will bind to the receptor and there will be an action generated, right? However, if acetylcholine builds up, acetylcholine esterase builds up, say suppose it is now in the proportion 2:1. Now there will be a greater chance of acetylcholine getting degraded by acetylcholine esterase compared to binding to the receptor. So what will ultimately happen is the action will be faded, right? If I
Yes,
continue
sir.
to build the concentration of acetylcholinesterase, then what is going to happen is that I'm going to mellow down the action which is required by acetylcholine on the receptor, right? Are you getting the point?
Yes, sir.
Yes, sir.
Can I not control this now? I can control this very easily by synthesis of acetylcholinesterase, right? And synthesis of this enzyme is under the control of the presynaptic and presynaptic neuron. So now imagine this situation. Now this is a condition what happens. Okay, have you ever heard about a disease which is called as Alzheimer's disease? It's called as AD in short. Alzheimer's disease. Has anybody heard about it? This is associated with dementia, progressive age-related progressive dementia. This is basically that when you age, then you stop your system stops to what you can say uh, remember the things. That means your brain stops remembering simple things, but long-term memories are still retained. That is because those synapses are quite tight. However, when you are learning something on daily basis, then you need to synthesize new synapses. You need to form new connections of the neuron. Then, if the situation is that you are heading to say Alzheimer's disease, then what happens is there is more concentration of acetylcholine esterase compared to the number of receptors. What that will do is that although your first neuron fires, the next neuron will not fire. Are you getting the point?
Yes, sir.
Okay, and that is essentially because of the building or building up of concentration of the enzyme acetylcholinesterase. Now, this is one way in which you can control the transmission of the nerve impulse, or continuation, you can say, of the nerve impulse. Okay. This is easy because in terms that acetylcholine synthesizing machinery is not that tough. That means you don't require much of a, a complex kind of an arrangement for synthesis of acetylcholine back. You can very easily take acetate, you can very easily take choline, and you can club them up together in order to synthesize acetylcholine back. So this is an easy stuff. However, there are certain neurotransmitters, like you might have heard about dopamine. If I need to synthesize dopamine, I will need at least 8 to 10 enzymes to synthesize dopamine. So, is it then logical that I use the same mechanism in order to dampen the effect which is created by dopamine release in the synaptic cleft? Is the question clear?
No, sir.
No, for what? Question is not clear.
Yes, sir. Question is not
Okay.
clear.
Okay. So understanding is this much is fine, that I can degrade acetylcholine and I can rely upon acetylcholine esterase because I can degrade it easily. I can synthesize it easily as well. Okay. So this logic fits good. However, if I need, say, 8 to 10 enzymes for synthesis of dopamine, which is another neurotransmitter, so can I work on the same mechanism to control the effect of dopamine on the postsynaptic membrane? That I also have some enzyme which can degrade dopamine. So once dopamine is degraded, then I will have to turn on all these 8 to 10 enzymes in my presynaptic neuron to resynthesize dopamine back. So does this logic fits here as well? That can I use a dopamine degrading enzyme in the synaptic cleft to control the action of dopamine? Is the question clear now?
Yes, sir. It is clear.
Okay, so what's your answer? Yes.
Sir, I think it won't.
Now, okay, this is something like I will give you an analogy. Okay, for acetylcholine esterase, is something like I have a 10 rupee note with me, and I give it to somebody and ask for change. So he gives me 5 rupee back, 5 rupee, 2 coins of 5 rupees. Okay, so it is essentially 10 rupees only. But if I want 10 rupee note back, then I can give 2 coins of 5 rupees again to some other person and get my 10 rupees back. So there is no net, what you can say, energetically, there is no net change happening. Is this much fine? This is good for acetylcholine, right?
Yes, sir.
Okay. But for dopamine, it's slightly different. Like imagine this, that how many of you would prefer that you give 1 rupee coin, 500 1 rupee coin, if you try and give it to somebody, will they be happy to give you a 500 rupee note?
No, sir.
No. The reason is not energetically favorable, right? Meaning you will have to spend more energy in dispersing those 1 rupee coins to get that essential value of 500 rupees, right? Are you
Yes,
getting
sir.
my point? Okay. So now if I fit that logic here, then degrading dopamine doesn't seem to be very logical, correct? To control its action.
Yes, sir.
Because to synthesize dopamine, I will have to activate, say, 8 to 10 enzymes. That's a very large price to pay, correct? So in what way then you will control the action of dopamine? Yes. So can you figure out some way by the way of which you can control the action of such neurotransmitters which are quite expensive to synthesize by the presynaptic neuron? Do you feel that this is a logical question to ask?
Yes, sir.
Okay. So can you try and answer?
So dopamine is made up of 8 to 10 different enzymes, right?
No, it is not made up of 8 to 10 different enzymes. You require 8 to 10 different enzymes to synthesize dopamine. It is not made up of 8 to 10 enzymes.
Okay, sir.
Okay. Yes, it's still a
Sir,
small molecule like acetylcholine, but to synthesize dopamine, you will require 8 to 10 different enzymes. That means it's a pathway for synthesis of dopamine. Something like if I write it like, say, A is converted into B, converted into C, converted into D, and so on and so forth, till you get dopamine. So there are precursor molecules, and there is enzyme 1, there is enzyme 2, there is enzyme 3, and so on and so forth, which are catalyzing this reaction of A conversion into B. So even if one is absent, then you cannot form dopamine. That's how it goes. So is there any way that you can think about that how you can control the action of dopamine? Or will it be like once it is released, it will continue to show you the action? Can you afford doing that?
sir, if one of the enzymes activity is degraded, then
No, then you will not synthesize dopamine. If you don't synthesize dopamine, what are you going to release?
yes, sir.
Yes. How will you control the action of dopamine? This is when it is already released in the synaptic cleft. How are you going to control its action? And why this control is required? Because if you don't control it, then this will continue to bind to the receptors and continue to initiate action potential in the next neuron. You don't want it, right? You want one action potential and that's done. It should cease out. Otherwise, this is something like you're sending an impulse from your brain, which tells your hand to move uh, an object from one place to another. If you don't stop this mechanism, then you will continue to move that object over and over again. Is it logical?
Yes, sir.
So what you do in order to control the action of dopamine like neurotransmitters? Any idea? Yes.
Sir, no idea.
Can you think about any way by the way of which it can be done? Now, there are several ways of doing it. One at least I am asking out from you people. This is the way that has been depicted here. This is the way about acetylcholine esterase action. It is degraded, so it can no longer initiate or open the channel.
Sir, there must be something which will degrade the dopamine in synaptic left.
See, degrading dopamine, meaning in the presynaptic neuron, you are going to again require 8 to 10 enzymes to resynthesize it back. That is what you don't want, right? You don't want that the dopamine that you have secreted out should get degraded. Because if you degrade it, then you will have to energetically spend more from your pocket in order to synthesize dopamine back. So every time your action potential arrives here, you're going to lose a lot of dopamine, right? Your cell
Yes,
body will
sir.
have to synthesize it over and over again. It is energetically not favorable. You will spend more energy in synthesis there. So any idea? Anybody can answer. Just I want to know how good you are in thinking the ways out. Yes. There has to be some way, correct?
Sir, something must bind to dopamine. Like,
Mm-hmm. And should not degrade it.
yes, yes. Means only bind and means it will form a competition for receptors. Like
Right.
that.
So I can do this one stuff. Good. I can do this one stuff that there should be a compound, say a dopamine inactivating group, an inactivator, which will compete with the dopamine to bind in competition with the receptors. That can be done. Now, what's the take here? Meaning, if I use an inactivator for dopamine, then what I'm ensuring that the dopamine doesn't get degraded. One, but if it doesn't get degraded, then I should be able to get it back in the presynaptic membrane, right? So that I can reuse it.
Yes, sir.
So if there is an inactivator, then it will bind to dopamine, but will it ensure that it comes back to the presynaptic membrane? You can extend this analogy a bit further. Imagine that this is a presynaptic membrane and this is the postsynaptic membrane. Now imagine that this is dopamine and this is a dopamine inactivator protein which binds to it. Imagine that you have something here which binds to the inactivator protein when it is bound to dopamine. It looks somewhat like this. Dopamine is here. So it is brought in close contact with something, some kind of a channel that can actually take up dopamine back. Can this work?
Yes, sir.
So instead of having inactivating group, can I not just have a channel that can take dopamine back? Yes,
Yes, sir.
I can
Yes.
have that, right? I can also have it. So this is what is given over here. There is a reuptake of neurotransmitter by means of a, what you can say, a transporter, which is a symport kind of a mechanism. Sodium ion gets in, and in response to that, the neurotransmitter is also taken up back. Okay, so this is an interesting stuff. Okay, I will just give you a highlight of this because we are almost reaching towards the end of the class. So I will just give you a highlight of this. What happens at the cell body? How is the neurotransmitter filled up in the synaptic vesicle? So there is a kind of an antiporter system where uh, protons are funneled out from the synaptic vesicle, and in response to that, the neurotransmitter gets in. And inside concentration, if you need to funnel out protons, then the inside concentration of the synaptic vesicle should be high for protons, right? So this is maintained by a proton pump, which is ATP guided. Okay. So now this is how the neurotransmitter is pumped inside the synaptic vesicle. Next is it is brought down now. This movement to the active zone means that you are actually transporting. This is an anterograde transport happening, where you are transporting the synaptic vesicle to the axon terminus. And now what happens at the axon terminus is that they are docked very near to the presynaptic membrane. Now this is a presynaptic membrane, and they are docked by using a docking protein. Okay, so they remain here. The synaptic vesicles filled with neurotransmitters, they remain here. Now, I didn't tell you what happens at the presynaptic membrane, right? So what is happening here is that in response to the action potential, that means in response to the depolarization of the plasma membrane, there is opening up of this channel, which is a voltage gated calcium ion channel. So now in response to that, in response to the action potential or in response to depolarization of the membrane, this voltage gated calcium ion channel opens. And now because there is an inflow of calcium, the incoming calcium binds to these docking proteins and changes their conformation. And because of that, they are moved apart. Now, previously they were together, but as soon as they bind to calcium, they move apart. And as soon as they move apart, the in contact with the membrane, the presynaptic membrane. And that causes exocytosis, meaning release of the neurotransmitter into the synaptic cleft. Now, this is what till now we have studied. Later, what happens is that these synaptic vesicles are recycled back in order to synthesize uncoated or empty vesicles, and even the neurotransmitter is taken back, where the neurotransmitter is filled up, is used to fill up the empty synaptic vesicle back at where? At the cell body. Okay. Now I hope I know that this is a bit tough for you to imbibe right now, but this is how the cycle continues over and over again. This is the process. We'll discuss this process in greater detail in the next class. So for time being, I feel that this is enough for you. So if there are any doubts in your mind regarding what we have done today, then you can ask me right away, or else we will meet again in the next class in order to